Bismillahirrahmanirrahim. This is the 10th video on the series of molecular bases of cancer. In this video, we will discuss invasion and metastasis. These are the references for this video. About 90% of the cancer patients die of metastasis. The metastasis and invasion is a multi-step process. The first step in this is the invasion of the basement membrane underlying the tumor. Then these tumor cells move through the extracellular matrix and penetration of the vascular or lymphatic channels occur. They survive within the circulating blood or lymph and they form a home at a new site, exiting from the circulation there and establish a micrometastasis site. Then this micrometastatic foci grow into sizable tumor mass. Normally the cell rests on basement membrane and this basement membrane separates the cells from the extracellular matrix, and these resting cells should become motile cells to invade local tissue as well as to move distantly, and this is achieved by the release of proteases which comes from cancer cells and also from the tumor associated cells such as tumor stromal cell or inflammatory cells which are associated with the tumor. The tumor cells become motile through invadopodia, and these invadopodia are basically actin cores and integrins. Through the release of or action of the proteases, there is lysis of the basement membrane first and subsequently the degradation of the extracellular matrix. The integrin, which is associated with invadopodia, serves as anchor and also promote invadopodia development, and it also guide the cell toward the stimulus or the chemotactic stimuli that results in its navigation within this area. So MMPs or matrix metalloproteases and other proteases function to achieve this important travel of the tumor. This flow diagram is summarizing the events of the mechanism of tumor invasion and metastasis. That is a multi-step process. The tumor that is confined to the above the basement membrane achieves its invasiveness by interaction and expression of many adhesion molecules. This tumor first sends many soluble mediators, and through these soluble mediators it forms a distant site that soils the fertile land for the seeding of the tumor. Then the tumor achieves invasiveness and it transits from epithelial to mesenchymal morphology, that is termed as epithelial to mesenchymal transition, and travel through basement membrane. This basement membrane is degraded. A basement membrane as well as the extracellular matrix is degraded by the action of various proteolytic enzymes which are derived from the tumor cells and also from the tumor associated cells, and these tumor cells then travel further and invade and penetrate the blood vessels and lymphatics. After survival in the blood vessels or lymphatics, the tumor now exists in the vascular lumen, and it establishes micrometastasis at the distant site. So it may remain dormant or reseeding can occur, and following dormancy there is a growth phase for these tumor cells. Again, this is the summary of the mechanisms, the multi-step process of the metastasis and invasion. In one, the tumor that was confined above the basement membrane achieves invasiveness by interaction and expression of many adhesion molecules. And now there is transition between epithelial to mesenchymal morphology that is termed as epithelial to mesenchymal transition, and now tumor cells travel through the basement membrane with the release of proteolytic enzyme from the tumor cells as well as from the tumor associated cells. Then the extracellular matrix is degraded by the action of proteolytic enzyme, and then there is traveling of the tumor cell further into the extracellular matrix, and it reaches to a blood vessel, and there it survive in the blood vessel or lymphatic. It is now present within the vascular system. It establishes a micrometastasis at the site where it leaves the vasculature. It exits as it entered the vessel through the degradation. It also exits through degradation of the through the vessel wall, basement membrane, and then extracellular matrix, and leave the vasculature in the destination or the destiny of this tumor. These micrometastases now grow into gross metastasis of metastatic tumor. For invasion and metastasis, it is essential to develop a metastatic clone of the tumor cells that travels further, as we have just discussed, and to complete its journey from the site of tumor to the destination or the site of metastasis, the tumor cell must undergo series of changes and also work in harmony with the tumor associated and extracellular cell matrix to survive. There are about 1 million cells they metastasize per gram of the tumor per day, but metastasis is not that much frequent. The reason being that the detached cells must overcome the host immunity and must be adaptable to the given microenvironment, which can be different from the original site of the tumor. The basement membrane and the interstitial tissue, the interstitial connective tissue, both make extracellular component, and the tumor cells are resting on the basement membrane, and underneath this basement membrane there is the interstitial connective tissue. So both these interstitial connective tissue and basement membrane are made up of different combinations of glycoprotein, proteoglycan, and collagens, and when this tumor cell is going to metastasize or going to invade the local structure, there should be degradation of the basement membrane first, and then the degradation of the extracellular matrix or the interstitial connective tissue. Then this tumor cell, the invasive tumor cell, now find the way toward the blood vessel and extravasate into the blood vessel, acquire a clone of the cells over there, and then they go to the distant site and extravasate into the destination. So the major steps for the invasion of extracellular matrix are loosening up the tumor cell interaction. That means the tumor cell is not now going to motile, degradation of the extracellular matrix, both the basement membrane and the interstitial connective tissue, and attachment of this tumor cell with the remodeled extracellular matrix components, and then migration and invasion of the tumor cells. For the initiation of this local invasion, the tumor cell should transform into a mesenchymal phenotype, as the epithelial cells are bound together by tight junction. They are not motile and they cannot invade. So these cell-to-cell tight junctions are lost, and tumor assumes a new phenotype, that is a non-polarized mobile mesenchymal cell. And this transition from epithelial to mesenchymal cell is reversible and it is temporary. So hypoxia is the major factor that is responsible for this epithelial to mesenchymal transition, as if the tumors are growing in epithelial lesions such as the intraductal carcinoma of the breast, there can be presence of central necrosis because of the rapid increase in size of the tumor and epithelial cells, and the diffusion radii for the glucose and oxygen are only about 100 to 150 micron. So the hypoxia inducible factor is increased in that area, and this hypoxia inducible factor is a transcription factor that is responsible for the degradation of the basement membrane and degradation of extracellular matrix through the transcription of matrix metalloproteases 2, matrix metalloproteases 1, and lysyl oxidases. The hypoxia is also responsible for the various other transcription factors such as slug, snail, twist, and ZEP1. The snail and twist downregulate the expression of E-cadherin, that is a glycoprotein that anchors the epithelial cells to each other and suppresses the motility. So when this is downregulated, the expression of E-cadherin is lost. That will result in the increased motility of the cell. And another, these twist and ZEP also, they downregulate the anti-proliferative proteins P16 and P21. Tumor is not only composed of tumor cell, it has got various associated cells that arise from the extracellular matrix, or some are recruited from the bone marrow. These tumor associated cells, they comprise about 50% of the tumor mass, and they include macrophages, leukocytes, fibroblasts, vascular endothelial cells, neuronal cells, and fat cells. E-cadherin are transmembrane glycoproteins that are responsible for cell adhesion and relay of intracellular signals. E-cadherin gene is mutated in gastric and prostatic carcinomas. This characteristic cell adhesion gene of the cell should be silenced for epithelial to mesenchymal transition, because now cell is transforming to a motile phenotype. This is controlled by transcription factors twist and snail by downregulation of the epithelial marker E-cadherin and upregulation of the mesenchymal markers such as vimentin and smooth muscle actin. The second step in the invasion of the tumor is degradation of the extracellular matrix. The tumor cell do so by the release of proteolytic enzyme or by inducing stromal cells such as fibroblast and the inflammatory cells. There are many proteases that are secreted in that environment. These are matrix metalloproteases, cathepsin D and urokinase plasminogen activator. And these matrix metalloproteases are important for remodeling of the basement membrane and remodeling of the interstitial connective tissue, and they are also important for release of the factor that contribute to the malignant behavior of the cancer. One example is matrix metalloproteases 9, that is also termed as gelatinase, and these gelatinases are responsible for cleavage of collagen type 4. That is an important component of epithelial and vascular basement membrane. The collagen type 4 cleavage product are responsible for chemotaxis, angiogenesis and growth promotion effect. The gelatinases are also responsible for release of vascular endothelial growth factor from the extracellular matrix sequestered pools, and the concentration of these MMP9 is increased in malignant tumors. The tumor cells are stimulated by nearby stromal elements, and they are responsible for the invasiveness of the tumor cells. In addition, they co-opt the function of a normal stromal cell, and they trigger inflammatory reaction and also recruit additional cell to that area, and hence they break the barriers that are present over there in that environment, and hence a tumor cell become a invasive cell. There are many cells which have been recruited by these tumor cells. These are the inflammatory reactions and others, but inflammatory cells are not here to protect the host. Inflammatory cells, in fact, they also secrete various cytokines and other proteases in that environment. The matrix metalloproteases are a family of endopeptidases that are regulated by tissue inhibitors of metalloproteases. These are secreted and synthesized by normal cell during tissue remodeling. Invasive and metastatic phenotype of the cancer cells are also associated with secretion of these matrix metalloproteases, but there is basically dysregulation between these proteases and its inhibitor, that are the tissue inhibitors of matrix metalloproteases. The invasiveness of a tumor is directly proportional to the increased matrix metalloproteases expression, while it is also directly proportional with the decrease of the inhibitors of these metalloproteases. These can be produced by the tumor cell as well as from the stromal cell, or can be produced by both. The metalloproteases are secreted by stromal cells, and they bind to the integrin on the surface of the tumor cell, and so that provide increased protease activity precisely where it is needed in order to invade. So that lead to the that enables this tumor cells to enter and travel through the extracellular matrix. The tumor cell motility is increased by increased expression of chemokine receptor type 4, and this tumor cell also affect the stromal cell, and from stromal cell there is another ligand for this receptor, that is SDF1 or chemokine ligand type 12 is secreted from the stromal cell that bind with this chemokine receptor 4. Hence the tumor cells have got the ability to invade. The marrow-derived suppressor cells are recruited from the blood and bone marrow, and they are responsible for the degradation of basement membrane and extracellular matrix by secretion of matrix metalloproteases. They also stimulate angiogenesis by secreting platelet-derived growth factor and vascular endothelial growth factor, and they are present at the edge of the developing tumor, and they also affect host response to the tumor. While the tumor-associated macrophages are present at the areas in which the basement membrane is breaking down, the urokinase plasminogen activator and cathepsin proteases are secreted by tumor-associated macrophages. The urokinase plasminogen activator converts the plasminogen into plasmin and helps digest the type 4 collagen that is present in basement membrane, while cathepsin proteases are secreted in response to the interleukin 4 from the tumor cells, and they also are associated with the lysis of the basement membrane. The carcinoma-associated fibroblasts, they secrete proteases that facilitate tumor cell invasion. They also secrete growth factors and angiogenic factors. They also secrete some factors that recruit precursor cells from the marrow, and these precursor cells become vascular endothelium later on. Adipocytes are also contribute in the tumor stroma, and they are also responsible for the crosstalk between the tumor cells, and they facilitate early stromal invasion by the malignant cell. The fat cell near the tumor often express a particular matrix metalloprotease that assist cancer cell in traversing surrounding connective tissue, and adipocyte-derived interleukin 6 stimulates tumor cell invasiveness. There is another product from the leptin that induces macrophages to secrete pro-inflammatory cytokines, which in turn promote invasion and metastasis, as we have seen earlier. So the contribution of the adipocyte is MMP secretion, interleukin 6 assisted function, and leptin function. The lymphocytes are also involved in this stroma cell interplay. The T cell may facilitate tumor invasiveness via tumor associated macrophages, and there is epidermal growth factor related activation in some type of cancer via the tumor associated macrophages, which have been activated by CD4 lymphocyte. Now this stroma can be termed as cancerized stroma because this stroma is influencing and influenced by various cytokines and factors. The epithelial to mesenchymal transition of the malignant tumor or tumor cell will liberate some stromal growth factor and also some chemokine, inflammatory cytokine, and immunomodulatory factors. The stromal growth factor will be recruiting the fibroblasts, the cancer-associated fibroblasts, and these cancer-associated fibroblasts are involved in angiogenesis as well as proteases release. Also, these cancer-associated fibroblasts, they recruit cells from the bone marrow, and these recruited cells are precursors for the endothelium formation. The cancer-associated fibroblasts are also responsible for the release of growth factor and various invasion factors. The other cells, such as the mesenchymal stem cell, the tumor-associated macrophages, the myeloid-derived suppressor cells, and the dendritic cells, they also liberate various angiogenetic factor and proteases, and also liberate growth factor, invasion factor, and immunomodulatory factors All these, they are responsible for the development of this cancerized stroma. Now the attachment of the tumor cell to the remodeled extracellular matrix. The third step in the invasion of the tumor involves changes in how tumor cell attach to the extracellular matrix protein. Integrin, that is a transmembrane protein, participate in adhesion of cell to cell adhesion and cell to extracellular matrix adhesion. In normal epithelial cell, integrin that binds basement membrane laminin and collagens are strictly restricted to basal area of the cell. These receptors, they help to maintain the cell in resting state, and loss of adhesion in cell will induce an special type of apoptosis that is termed anarchies. Anarchies literally means homelessness, and this occur in detached normal cell. But the free tumor cell has got resistance to this form of cell death because of expression of other integrin that reduce the loss of adhesion to extracellular matrix, apparently by transmitting signal that promote cell survival. Moreover, the stroma, as we have seen, is cancerized, and it is also promoting invasion and metastasis. Now this detached cells in the background of an altered or cancerized extracellular matrix, the cell is propelled forward through the degraded basement membrane and the proteolized matrix. The cell attached to the matrix at their leading edge while detached at the trailing edge. Hence they move forward. They bind or attach at the leading edge and detaches from the trailing edge. So the contraction of actin cytoskeleton will move it forward. This is occurred by autocrine motility factor also. The factor that stimulate and direct movements are tumor cell derived cytokines, chemokines and growth factors, the cleavage product of the matrix component, and stromal cell derived paracrine factors, hepatocyte growth factor, scatter factor. These paracrine factor bind to the receptor tyrosine kinase on the tumor cell and stimulate motility. The invading cell make the environment favorable by the changes made into the extracellular matrix and the secretion of various factors in the environment. The motto of these malignant cells is to acquire access to vascular space, which is the initial step in the metastasis. During its journey within this extracellular matrix, the tumor cell interact with the extracellular matrix cells, the innate and adaptive immune cells, the fibroblast and the endothelial cells. When the tumor cell successfully enters the circulation, these are prone to destruction by mechanisms such as mechanical shear stress, anarchies, and innate and adaptive immune defense mechanism. However, the viable circulating tumor cells are present within the circulations in case of solid tumors. So, which cell metastasize and what decides about their destiny and route? These are still under investigation. Some clues are identified. One is that the malignant cell, they move in groups. The clumping is facilitated by homotypic interaction, that is among the tumor cells, or this clumping is also facilitated by the heterotypic interaction, and this heterotropic interaction is between the tumor cell and the elements of the blood, especially the platelets. These interaction or clump formation increases their chances of survival within the circulation. The tumor cell embolus is stabilized by expressing polyphosphates that are anionic substances, and this result in activation of contact factor, the factor 12, which result in fibrin deposition. Hence the tumor clump is stabilized. The clump of cells may contain some tumor cells with stem cell-like property, which may contribute to unrestricted growth and plasticity of the tumor cell, and these characteristics favor malignant cell to survive and to flourish in the new microenvironment. Now the tumor has invaded the surrounding tissue, and it should now get access to lymphatic or vascular channel. To invade the circulation, the tumor cell need to pass through the wall of the vascular or lymphatic channel. These tumor cells can be in two forms: the solitary tumor cells and the clusters of the tumor cells or clumps of the tumor cells. The solitary cells that have acquired mesenchymal phenotype through epithelial to mesenchymal transition is only a small part of the primary tumor. They get access to the vascular or lymphatic channel lumen more rapidly than the clusters. The vascular intravasation is made more favorable because the tumor associated vessels are not completely initiated or protected by parasites. Moreover, the vascular endothelial growth factor that is present in this environment also increases vascular permeability. This leakiness is further increased by matrix metalloproteases and other proteins and stromal cell factor. Tumor associated macrophages produce epidermal growth factor and colony stimulating factor 1 that also facilitated this process of intravasation by these lonely cells. The other form of the tumor cell is the clusters of cells or the clumps of the cells, and they prefer to go to the lymph node and stay there for some time. Later on, through the venous and lymphatic connections, they get access to the systemic circulation. This journey to the lymph node is not through blood vessel, and then these two pathways also differ for various tumor types. Some tumor types they prefer to disseminate through lymphatic channels, and some tumor types they prefer to disseminate through vascular channel. The metastasis can remetastasize to other sites also. Currently, the SS for single tumor cells has got important prognostic and therapeutic implication. They are successfully used as a guide for choice of chemotherapy. Now the aim of the circulating tumor cell is to find a board and to survive in the environment. So the diameter of this circulating tumor cell is about 20 to 30 micron. So it should find a home for itself before filtering out by mechanical shear forces in the lung capillaries, where the diameter of these capillaries is about 8 micron. So when they pass through the pulmonary circulation, they will be filtered out from the blood. Then The individual cells are prone to anarchies, that is the special form of apoptosis when cell detaches from its attachment. So to avoid anarchy, one mechanism has been recognized, that is the activation of track B. This track B or tyrosine kinase B mechanism activation is a suppressor of IKs. So these tumor cells are finding abort, and this abort is not livable. So they issue pay order to the bank, and bank releases currency for the construction of this abort. The bank in this example is bone marrow, from where the hematopoietic progenitor myeloid cells are secreted, and they reach to their destination for the formation of this abort or the pre-metastatic environment or niche. So the tumor cell, they issue checks that are vascular endothelial growth factor, placental growth factor, and the various other growth factors that act on bone marrow, and from bone marrow bank the currency is released, that is hematopoietic progenitor myeloid cells. These hematopoietic progenitor myeloid cells, they are responsible for the development of pre-metastatic niche or the construction of this abort for the tumor cells that has started in the specific organ even before the start or local invasion of the tumor itself. So this is achieved by cytokines and enzymes that are derived from the hematopoietic progenitor myeloid cells, and also from the inflammatory cells that are recruited from the organ stroma and also from the blood. The inflammatory cells release lysyl oxygenases and metalloproteases that are important for the construction of this abort for the tumor cells. This perimetastatic niche undergo repeated remodeling by the cytokine and enzyme that are produced by these sources. Now, what is the mechanism of tumor cell arrest within the circulation? This is important because this association of tumor cell with the constituents of the blood, formed elements of the blood such as neutrophil, immune cell or platelets, is important because it protects the tumor cell from sheer mechanical forces, immune reactions and other stresses while it is present in the circulation. So there is an important glycoprotein expressed at the surface of the tumor surface, that is the CD44, and this CD44 actually binds with the P-selectin or E-selectin, and this adhesion is stabilized further by the expression of a factor that activates platelet via production of thrombin, and this activates fibrin and wall Willebrand factor, and through interaction of the one Willebrand factor, fibrin and integrin, there is a stable adhesion between these platelet and the tumor cell. So the binding of the tumor cells to platelet require P-selectin and the platelet integrin that is responsible for the recognition of CD44 via von Willebrand factor or fibrin. Tumor cell also bind with the leukocyte or with the endothelial cell membrane through CD44 and other selectin ligands that are the P or E selectins. Now the vessel wall has got the hold of the tumor cell and its fellow travelers, but the anatomy of the blood vessel at the destination from where these tumor cells are going to extravasate is different. Here the blood vessel is complete, and it's not like that of the tumor blood vessel where the anatomy of the blood vessel was incomplete. It was not completely invested by parasites. Here it is complete. It has got complete investment by the parasites. This extravasation mechanism is organ specific because there are proteins that increase the vascular permeability, that degrade tight junction between the endothelial cell and promote tumor cell escape from the circulation. So various factors are involved in the extravasation of one tumor type into the one organ, one specific organ, and these may be different for other organ metastasis or extravasation. For lung, the important proteins that are involved in extravasation of the tumor cells are VEGF, MMPs, the vascular endothelial growth factor, the matrix metalloproteases, cyclooxygenases, and the epiregulin, that is the ligand for epithelial growth factor receptor, epidermal growth factor receptor, and then there is another important protein that is responsible for the dissolution of the tight junction between the endothelial cell, and this is angiopoietin-like 4 protein. The circulating tumor cells, when forming a colony at a distant location, should synchronize with the biochemistry, the cellular composition of the matrix, and the extracellular matrix itself. And these metastatic foci or the circulating tumor cells may survive, but they never proliferate, and they may die. So there are many challenges which are faced by these circulating tumor cells to establish this distant colony, but it has to overcome all these challenges, and there should be inadequate blood supply and the suitability of the extracellular matrix as well as the composition of the extracellular matrix to develop a micrometastatic site. Most of this is achieved through the HPCs that have been recruited through the blood vessel, that have been recruited from the bone marrow, and these HPCs, progenitor cells, hematopoietic progenitor cells that have been recruited from the bone marrow, they express SDF. This SDF protein binds with its receptor that is present on circulating tumor cell, that is the chemokine receptor type 4, and it is responsible for the proliferation of these tumor cells and development of micrometastasis. For the continued survival of this micrometastasis, SRC signaling is important. That is a non-tyrosine kinase that transduces signals that are involved in the control of a variety of cellular processes such as proliferation, differentiation, motility, and adhesions. Circulating tumor cells are present in large quantities in the cancer patients, and there are about 1 million tumor cells are released per gram of the tumor per day. They are detectable even before the onset of the pathologically invasive metastasis, and the reason for the failure of establishment of micrometastasis despite having large quantity of these circulating tumor cells are not clear, although it is considered that there are many factors that are involved in the inefficiency of the metastasis, and these are some inherent factor within the tumor and some inherent factor that are present in the stromal cells and cell signaling. What is the fate of these circulating tumor cells? They may die, they may become quiescent, or they may form a gross malignant tumor. So the mechanisms that are involved in the death of these circulating tumor cells are apoptosis, anarchy, immune elimination, or other mechanism, while dormancy is a state that can be defined as presence of cancer, whether primary or metastatic, and that do not enlarge to a point of being clinically detectable. So the cancer is there but is not detectable clinically, and there is variable duration for various type of the tumor, and these dormant tumors may become focus for the micrometastasis, and the mechanisms that are involved in the escape from this dormancy or somnolence are angiogenesis, immune surveillance, apoptosis after oncogene inactivation, inflammation, activation of cancer stem cell, and activation of adhesion molecules such as integrins. In instances, the proliferation of the cell and the death of the cell is in equilibrium, so the amount of the or number of the cell that are being proliferating is equal to the numbers that are died. So there is constant equilibrium between these micrometastases, so there is no gain of size of the tumor, and this may go into dormancy, and these cells are into G0 phase of the cell cycle, and it is considered that this is due to involvement of various metastasis suppressor genes, and due to this concept, after resection of the tumor, adjuvant therapy is given to the cancer patient for the undetected micrometastasis. Such treatments are of short duration. This illustration is describing the fate of the tumor cells that have got the or formed the micrometastasis. The primary tumor may form a gross metastasis, or they may die due to apoptosis or due to therapies such as radiation, chemotherapy, or surgical removal, or this primary tumor may develop micrometastasis that remains in quiescent stage, and in this stage the cells are in the G0 phase of the cell cycle. Then there are some factors that are responsible for the activation of the cell cycle, and they push this cell from G0 phase to the proliferation, and a gross metastasis is developed. Similarly, the balanced proliferation of the cells and their death, if imbalanced, there will be excess of cell proliferation. Three factors are related to the homing and proliferation of the circulating tumor cells. So these are the location and vascular drainage of the primary tumor, tropism of particular kind of the tumor cell to the specific tissue, and the escape from tumor dormancy. The location is described, and it is described that the tumor metastasizes according to the venous or blood drainage of that particular organ, such as the CA colon goes first to the liver, and from there it goes to other sites. However, it is seen that there are some exceptions to this rule, such as the carcinoma of the prostate and breast, they go to bone, and bronchogenic carcinoma goes to adrenals and brain, and neuroblastomas to liver and bone. This gives clue that there are some other mechanisms also responsible for this homing of the circulating tumor cell. So now the organ tropism is made possible by the expression of adhesion molecule whose ligand are found on the endothelial cells of the target organ. One of these is CD44 adhesion molecule that is expressed on normal T lymphocyte and is used by these cells to migrate to selective sites in the lymphoid cells. Such migration is accomplished by the binding of CD44 to hyaluronate on high endothelial cells. Solid tumors also often express CD44, as we have seen earlier. Then there are some cells, some cancer cells that express chemokine receptors, and these chemokine receptors are responsible for guidance of the tumor cell to the tissue expressing chemokine. So they are the guiding antennas for the cancer cells. Another point or concept is the seed soil hypothesis that was presented by Paget in 1889. He stated that tumor-associated stromal cells, in fact, they plow the road for the tumor spread to particular site, because a significant amount of blood is supplied to skeletal muscle and spleen, but these organs are not frequently, they do not show frequent metastasis to these sites. So these sites are non-permissive, they have non-permissive environment for the tumor, so they are unfavorable soils. Then there is another factor, that is escape from tumor dormancy. As we have seen that the metastatic cell fail to grow even when they take root and survive within distant tissue, such as in melanoma, breast and prostate, and in these instances the tumor cell secretes cytokines, growth factor and extracellular cell matrix molecules that act on the resident stromal cell, and these resident stromal cell in turn make the metastatic site habitable for the cancer cell. For example, in breast cancer, the metastatic foci or group of this cell to the bone secrete parathyroid hormone related peptide. This stimulates osteoblasts to express more rank ligand, and this activation of the rank ligand will activate the osteoclast, and osteoclasts now are responsible for the degradation of the bone matrix, and they also release growth factor within that are embedded within it, such as the insulin growth factor and the transforming growth factor beta, and these factors bind to receptors on cancer cell. Activating signal pathway are then activated, and these support the growth and survival of the cancer cells. As the development of angiogenesis within the tumor and its metastatic foci, they are responsible for the tumor growth. This can be limited by suppression of the tumor angiogenesis. This can be achieved by extracellular matrix and other angiogenesis inhibitors, and they are responsible for suppression of angiogenesis. One is the thrombospondin or TSP1, that is derived from a large extracellular matrix glycoprotein, and it is inhibitor of blood vessel formation, thrombospondin 1. Then angiostatin, this is a breakdown product of the plasminogen, and there are numerous fragments of the extracellular matrix constituents such as endostatin, inhibin, and many other. They restrain tumor-related blood vessel growth as well. P53 upregulates thrombospondin 1 expression, and so when it expresses the thrombospondin 1, so it is a strong anti-tumor angiogenic activity. SIRT or sirtuin deacetylase are important in stress responses and also in longevity, and one of the sirtuin, that is SIRT3, is it will increase the mitochondrial antioxidant level, that is manganese superoxide dismutase, and this will produce less reactive oxygen species, and when there is less reactive oxygen species stress on the cell, the hypoxia-inducible factor 1 activity is decreased. So hypoxia associated or inducible factor we have seen was responsible for angiogenesis. When it is decreased in concentration, so the angiogenesis is also inhibited. The body has the protective mechanisms or defense mechanism to prevent cancer from arising. It has also got protective mechanism that impedes the invasion and metastasis of the tumor. There is a suppressor activity for about every step of the metastasis and invasion, and this metastasis, so it is different. Metastasis inhibitor is different from the tumor suppressors. Tumor suppressors, they stop the initiation and progression or growth of the tumor, while these are limited to the metastasis, the inhibition of the metastasis. A metastatic suppressor is a molecule that must impede one of the invasion or metastasis related process without necessarily affecting growth and survival of the primary lesion. The nature of these metastatic suppressor is protein or microRNAs. About 30 metastatic suppressor protein have been identified, and there is a family of microRNA that has also been recognized as having function of the tumor suppressors. These tumor suppressors have got diversity in their mechanism of action. Some act multiple steps or only one. Some molecules may inhibit certain process in some tumor types, while they have got opposite effect in the other type of the tumors. Some metastatic suppressors have additional separate activities that are directed against the primary tumor. The epithelial to mesenchymal transition is an important step in the invasion of the tumor. This epithelial to mesenchymal transition is impaired by a number of factors. One of these is cadherin, that is the family of cell-to-cell adhesion molecules. The important one among these is E-cadherin, and it is expressed on the surface of epithelia, and it mediates cell-to-cell adhesion by mutual zipper interaction. Catenins, which can be subclassified as alpha, beta, and gamma, these proteins, these are the proteins that interact with the intracellular domain of the E-cadherin, and they create a mechanical linkage between the molecule and the cytoskeleton that is essential for effective epithelial cell interaction. Expression of both of these, the E-cadherin and catenin, is reduced or lost in most carcinomas, and it is downregulated by the transcription factor SNEL and ZEB1. The microRNAs are also responsible for the impairment of the epithelial to mesenchymal transition. One is the microRNA 101 and the microRNA 200 family. They help to maintain the epithelial phenotype. MicroRNA, they repress ZEB1 and ZEB2 level, thus they relieve their repression of E-cadherin level. The ZEB is also down-regulated by microRNA 200. The transforming growth factor, that is an inhibitor of tumorigenesis, is also a promoter of metastasis. This function is achieved by downregulation of microRNA 200s. In most carcinomas, the loss of E-cadherin is linked with development of an invasive and aggressive type of the tumor. And as we have seen in the video of the tumor suppressor genes, the beta-catenin also binds to the APC gene product. This effect is independent of its interaction with E-cadherin and beta-catenin and alpha-catenin. Mutation in either the APC or beta-catenin gene are linked in the development of colon cancers. The first tumor suppressor recognized is NM23H1. It inhibits tumor cell motility by blocking cellular mobility signals by ROS-related cell activation pathway. Another member of this tumor suppressor is P63. It is a member of P53 family and helps to restrain cellular invasiveness. P63 is expressed in some in situ carcinomas of the breast and prostate. It is often repressed or lost in aggressive metastatic carcinomas. Mutants of P53 may bind and inactivate P63 by forming heterotetramer aggregates. P63, acting as a transcriptional regulator, also upregulate expression of certain genes that inhibit metastasis, for example microRNA 130B. Now the movement through the connective tissue is an important part of the metastasis and invasion. This activity, it depends on integrin alpha 5 and Rho-A, and these are blocked by microRNA 31. The suppressors of introversion. The notch is a key inhibitor of tumor angiogenesis. The mechanism that impede introversion also involve notch. A protein that is called amino terminal enhancer of split (ICE) help to inhibit migration of tumor cell through vascular wall via signaling network that include notch activation. And there are some mechanisms that limit tumor cell survival within the circulation. One we have seen in detail, that is anoiky, and many endothelial cell express the Duffy blood group glycoprotein that is dark, and upon recognition Chi1 on tumor cell membrane, dark triggers senescence program. So this is termed as dark senescence. Cell of innate immunity also triggers cell death via TRAIL and CD95, and in some tumor, large size of the tumor cell will result in the release of nitric oxide by the cell lining the liver sinusoids, and this trigger apoptosis in the tumor cells. The microRNA 31 that inhibit the tumor cell invasiveness or intravision, it also blocks the extravision. This microRNA target both integrin 5 alpha and Rho-A in the process of the extravasation. Then there are some inhibitors of metastatic colonization, and this metastatic colonization may be the major limiting step in the tumor metastasis. KIS1 and its receptor KIS1R. This has got its name from the famous chocolate from the Pennsylvania, that is KIS, and this KIS1 that is made by the tumor cell binds to its receptor KIS1R that is present on cell membrane, and it results in tumor cell apoptosis. The other suppressors of metastatic colonization are also recognized. One is GATA3 that is present in breast cancer, and it promotes cellular differentiation and impedes multiplication. PSAP is seen in prostatic cancer, and it induces stromal cell production of the anti-angiogenic substance, that is thrombospondin 1. MicroRNA plays its role over here also. Multiple metastatic activities inhibit the ability of the cancer cell to colonize distant sites effectively.